Imagine being able to peek behind the curtain, into the tiny world all around us, revealing secrets our eyes normally miss. It's a pretty cool idea, right?
Definitely. And today, we're diving deep into an innovation that makes that surprisingly achievable.
Yeah, we're talking about a new kind of microscope built on open source principles, which is always interesting, and it has some really remarkable capabilities.
Absolutely. Our main source for this deep dive is an audio recording, ImHorPhen 1.m4a. It's a really engaging conversation, gets right into the nuts and bolts of how this microscope was developed.
So our mission today, for you, our listener, is basically to unpack this technology. We want to understand together how this clever instrument opens up a world we usually can't see.
Sounds good. Ready to explore what's hiding right under our noses.
Let's do it.
Okay, so let's start with a core innovation. They call it the InScope. The basic idea is, well, pretty ingenious.
Go on.
They took a standard 3D printer, the kind that builds objects layer by layer,
Right.
and they replaced the extrusion nozzle, the part that spits out the plastic, with a specially designed optical head.
Okay, hold on. A 3D printer turned into a microscope. That's unexpected. What's actually in this optical head?
It's quite packed, actually. You got a high-quality HQ camera, an Arduino Nano board that's for controlling the lighting really precisely.
Okay.
Then there's a ring of 16 LEDs, which is key for different illumination techniques we'll probably get into, and of course an objective lens.
16 LEDs.
Yeah. And what's really neat is that the original design for this InScope was open access, open source.
Love that. Open source for microscopy. But
Yeah.
how does just putting a camera on a printer arm let you see more than a regular microscope?
That's where computational microscopy comes in.
Right.
It's not just about the lens anymore.
Right.
They use computing power to squeeze way more information out of the images they capture. Specifically, they developed techniques for multi-angle and multi-focus imaging.
Multi-angle and multi-focus. Sounds advanced. Can you break those down for us?
Sure. For multi-angle imaging, the idea is to light up the sample with each of those 16 LEDs one by one, sequentially.
Okay, like taking 16 slightly different pictures.
Exactly. An image is captured for each individual LED illumination. Now, initially, they ran into some issues with uneven lighting intensity.
I can imagine.
So to fix that, they used a method called minimum intensity projection with ImageJ software. Think of it like taking those 16 shots and only keeping the darkest parts from each to create a final, more evenly lit image.
Clever workaround. And what about multifocus? Because, with microscopes, usually only a tiny slice is sharp, especially if the sample isn't perfectly flat.
Precisely the problem it solves. With multifocus, they capture a whole series of images while moving the camera up or down along the z-axis.
So changing the focus depth.
Yes, in very small steps, like 0.1 millimeters, you end up with a stack of images, like a digital flipbook, through the sample's depth. In that stack, different parts of the sample will be perfectly sharp in different image slices. The multifocus software then merges all the sharpest bits from across the entire stack into one single image.
Ah, I see. So the whole 3D structure appears sharp, even if it's bumpy or uneven.
Exactly. You're no longer limited by that super thin plane of focus you get with traditional optics. It really opens things up for looking at 3D structures without, physically slicing them.
That's really impressive. But the recording mentioned a limitation at first, something about focusing.
Yeah, good point. With that first HQ camera they used, the focus had to be done manually.
Like twisting a knob.
Pretty much. Adjusting the camera to sample distance until it looked sharp. It can be a bit slow and subjective too.
Right.
Which is why they then developed an algorithm to automate the focusing.
An algorithm that focuses by itself. How does that work?
It looks at the image and calculates how sharp it is based on 3 criteria. There's flatness, basically how uniform the focus is across the image entropy, which measures the amount of detail, and the variance of the gradient, which looks at how strong the edges and contrasts are.
So it's mathematically measuring sharpness.
Essentially, yes. The algorithm nudges the camera in tiny steps along that z-axis, calculates this sharpness score at each position using those criteria, and keeps moving as long as the score improves.
And when the score starts to drop?
It stops. It figures it's found the sweet spot, the optimal focus point.
Ingenious.
Oh.
Going from manual fiddling to automatic focusing must be a huge time saver and probably more consistent too.
Definitely, but the evolution didn't stop there. The recording talks about the underdecam.
Right, the next version.
Exactly. The underdecam is basically an improved InScope. The main motivation was to get that autofocus capability built right into the camera itself.
Ah, so they swapped out the camera?
They did. They replaced the original HQ camera with an Articam HQ model, which has built-in autofocus hardware and software.
And what are the practical advantages of that switch?
While mainly it's simpler to use and more adaptable. This new camera has a really wide focusing range from about 8 centimeters all the way out to infinity.
Wow, that's versatile.
It means it can handle a much bigger variety of sample sizes and shapes without needing complex manual adjustments. The recording also mentions they had to 3D print some modified parts to fit this new camera onto the optical head.
Again, showing the flexibility of that 3D printer base and the open source approach, iterating quickly.
Precisely. It allows for rapid improvements, often community-driven, leading to more robust solutions faster.
And there was also mention of using this system in controlled environments, like for plant experiments.
Yes, that's a really interesting potential application. Imagine putting an underdecam inside a growth chamber.
Right.
You could automatically monitor how plants react to different conditions, pesticides, temperature changes, different lighting in real time, getting incredibly detailed visual data. Huge potential for agricultural research.
Let's talk about actually controlling the whole setup. The recording mentions a Raspberry Pi 4 and G-code.
Right. The Raspberry Pi 4 acts as the brain, the mini computer running the show.
Okay.
It controls the 3D printer's movements, telling the stage where to go, and manages the other components like the camera and lights.
And G-code, isn't that what 3D printers use?
Exactly. It's the standard programming language for instructing CNC machines, including 3D printers, on how to move very precisely. So they use G-code commands sent from the Pi to position the camera accurately over the sample.
Got it. And what about image resolution? How good are the pictures these cameras take?
The original ENDERSCOPE camera could theoretically go up to 64 megapixels, which is quite high.
Wow.
But they mentioned running into limitations with the specific Python library they were using at the time, PyCamera 2. It apparently couldn't access that full resolution, so they were working with something lower in practice with that setup.
Ah, common software bottleneck. Okay, let's move on to the automated scanning techniques they developed. First up was raster scan or snake scan.
Right, the raster scan or snake scan because of its back-and-forth snaking pattern.
Yeah.
It's a systematic way to cover an area. The camera moves along a predefined path, going line by line, and snaps pictures at specific X and Y coordinates.
Methodical.
Very. But it can be time-consuming, especially for large areas at high resolution. And it generates a lot of image data.
Which leads us to stitching or Fusion 2D. What problem does that solve?
Well, when you do that snake scan, the images you capture inevitably overlap quite a bit at the edges.
Right, to make sure you don't miss anything.
Exactly. Stitching, or 2D fusion, is the process of computationally finding those overlapping regions, aligning the images perfectly, and then blending them together to create one large, seamless mosaic image of the entire scanned area. Without it, you just have a pile of individual overlapping photos.
Okay, so it pieces together the puzzle. Then they talk about a smart scan manual. What makes it smart?
A smart scan manual is a step up in efficiency. It starts by taking a quick, low-resolution overview image of the whole sample.
Like a map.
Sort of, yeah. Then, the user looks at this overview map and can simply click on specific objects or areas they find interesting. And the printer stage automatically moves the camera directly to that selected spot to capture a detailed high-resolution image just of that feature.
That saves a ton of time compared to scanning the whole thing in high res if you only care about a few spots.
Exactly. Much more targeted.
And there's a fully automated version too, the Smart Scan Automatic.
Yes. That's the next level. Once it's set up, it runs entirely on its own. No human clicking involved.
How does that work?
It also starts with that global, low-res overview image. But then, instead of waiting for a user, it uses image processing techniques to automatically detect objects of interest within that overview.
Okay, how does it detect them? What are the steps?
There's a sequence. First, the color image is converted to grayscale. Then a Gaussian blur is applied to reduce noise, sort of smooth things out a bit. Then it calculates the image gradient to find edges where brightness changes sharply. A binary threshold is applied to basically separate the potential objects from the background, making it black and white.
Okay, simplifying it.
Yeah. Then a morphological closing operation helps fill in any small gaps or holes within the detected object shapes. Finally, it finds the contours, the outlines of these shapes, and calculates their geometric center point.
So it ends up with a list of coordinates for the centers of all the detected objects.
Exactly.
But then how does it decide which object to image first, second, third, and so on efficiently?
That's where a classic computer science problem comes in. The traveling salesperson problem, or TSP.
The one about finding the shortest route to visit a bunch of cities?
That's the one. In this case, the cities are the centroids of the detected objects. The TSP algorithm calculates the distances between all pairs of objects and figures out the shortest possible path for the camera to travel to visit and image each object just once.
So it optimizes the camera's movement. Very clever.
Yeah, they mentioned using the Hungarian algorithm as part of their TSP solution to help minimize the travel cost or distance. It's like programming the world's most efficient delivery driver for the microscope.
Fascinating application. But the recording also mentions issues with geometric aberrations. What are those?
Geometric aberrations are basically distortions in the image that make things look misshapen. They identified two main types: radial and tangential.
Okay.
Radial distortion is often caused by imperfections in the camera lens itself. It makes straight lines appear curved, either bowing inwards like a pincushion or outwards like a barrel.
I think I've seen that effect in photos.
Yeah, it's common. Tangential distortion can happen if the camera sensor isn't perfectly parallel to the stage or lenses are slightly misaligned. It makes objects appear tilted or stretched in one direction.
Right, like looking through a slightly warped window.
Kind of. The point is, these distortions mess with accurate measurements and observations. So they developed an algorithm specifically to correct for both radial and tangential distortions, making the final images geometrically accurate. Crucial for scientific work.
Definitely. Now, a really key aspect of this whole project seems to be about making microscopy more accessible, democratizing it. How are they doing that?
A big part of it is the development of a user-friendly Python library and a graphical user interface, or GUI.
Okay.
The goal is really to open up this in-scope and under-decam technology to a much wider audience, people who maybe aren't expert coders. It's about lowering the barrier to entry.
Building on that open source spirit, not just giving away the plans, but making it easier to use.
Exactly. It accelerates innovation when more people can easily experiment and build upon the work. It leads to faster progress, more robust solutions.
So this Python library, it's like a toolkit for controlling the hardware. How is it structured?
It's designed to be modular, which is smart.
Yeah.
There are different software classes or modules for specific jobs. Well, there's a class for handling serial communication, talking to the hardware components. Another for controlling the stage movements, X, Y, and Z axes. One for the user interface, specifically designed to work well within the Jupyter Notebook environment, which is popular in science.
Okay, makes sense.
There's also a class for controlling the LED ring light via the Arduino, and another one for generating those scan patterns we talked about, like the snake scan.
And presumably, they provide examples to help people get started.
Absolutely. They've created a bunch of demonstration scripts, demos, that show how to use the different parts in the library.
Like mini tutorials.
Pretty much. Covering things like light control, stage movement, using the GUI, basic camera control, putting it all together for simultaneous control, running scan patterns, using the autofocus, capturing multi-angle images, and even running the smart scan routines.
That sounds comprehensive.
And critically, the library and all these demos are available open source on GitHub. It really encourages people to jump in, use it, adapt it, contribute back. It makes experimenting and adding new algorithms much, much simpler.
That's a fantastic initiative. Okay, finally, the recording touches on a really interesting extension, adding spectroscopy with the under-decamp spec. What's that about?
Right, the under-decamp spec is the latest evolution mentioned. It combines the under-decam's microscopic imaging with near-infrared, or NIR, spectroscopy.
So adding another layer of information.
Exactly. They integrated an infrared spectrometer probe with the Articam camera, still controlled by the Raspberry Pi.
And what does near-infrared spectroscopy tell you that regular imaging doesn't?
Spectroscopy, in general, analyzes how light interacts with matter based on wavelength. NIR spectroscopy is particularly good at revealing information about the chemical composition and physical properties of a sample.
How?
By measuring its reflectance spectrum, how much light it reflects back at different NIR wavelengths. With the under-decamp spec, they can scan an area and at each point capture not just an image pixel, but a whole reflectance spectrum.
So for every point on the sample, you get its unique light signature in the infrared range.
Precisely. Think of it as adding a chemical fingerprint dimension to the visual image.
That sounds like hyperspectral imaging. How does it work in practice?
Yes, that's the term. They scan an area, maybe using the snake scan or even a smart scan pattern. At each X, Y position, they acquire the NIR spectrum. This builds up what's called a hyperspectral cube.
A cube.
Yeah, you have the two spatial dimensions, x and y, like a normal image, but then a third dimension representing the wavelength and the reflectance value at each wavelength.
Got it. So you can analyze the spectral signature of anything within the scanned area.
Exactly. The recording gives a great example using a fern whiff.
Ah, yes. Tell us about that.
They use the underdecamp SMEC to study the water content in a fern leaf as it dried out over time.
How?
By measuring the reflectance in a specific near-infrared range between 1,300 and 1,600 nanometers, they observed that as the leaf lost water, its reflectance in that range actually increased.
Interesting. And they could verify this.
Yes. To validate their experimental results, they used established theoretical models for plant spectroscopy, specifically Purosale and Prospect, which are available as Python libraries.
Okay.
These models allowed them to simulate the expected relationship between water content and NIR reflectance, and the simulations matched their experimental observations really well. They also mentioned needing a calibration step, using a reference sample to ensure the measurements are accurate.
So you could potentially use this to monitor plant stress or health non-invasively.
Absolutely. Imagine using it to check the hydration of crops in a field, or even monitor your houseplants in a much more scientific way.
Yeah,
It's a powerful demonstration of the potential.
it really is. Okay, so to kind of wrap things up, we've journeyed from a modified 3D printer, the InScope, leveraging open source ideas.
Who add in clever computational techniques like multi-angle and multi-focus imaging, automated focusing.
Then evolving into the underdecam with its integrated autofocus for easier use, and developing smart scanning methods to be more efficient.
All made accessible through user-friendly Python libraries and GUIs.
And finally, culminating in the underdecam spec, which adds the analytical power of near-infrared spectroscopy to the mix.
It's quite an evolution. What really stands out is that potent combination of open-source hardware, advanced computation, and now spectroscopy. It genuinely lowers the barrier for accessing really powerful scientific tools.
It really does. And that leaves us with a final thought, doesn't it? What incredible discoveries might come about when tools like this, so accessible, so customizable, get into the hands of citizen scientists, educators, maybe researchers in places with fewer resources.
You have to wonder, the democratization of advanced instruments like this, it could really change how and where science happens.
Food for thought indeed. Thanks for joining us on this deep dive into the world just beyond our sight.